You're right, it's not. Thank you. All right, I'm not sure where everyone else is, but it's time to begin. So let me share my screen. Thought I had done that. Make sure I'm recording. Yes, there's the dot there. All right, today is October 31st, and we're supposed to be covering Lab 7, throat and urine cultures. Hopefully, either during this lab or after the lab, I'll get you the first results for the unknown project. Hopefully, I'll get that out. Any questions about what we're doing? All right, if not,
Not yet.
let's go on to Lab 7. Why isn't that at the top, or is that just blocking? There it is. All right, throat and urine cultures. There is some reading to do before you do the lab, so make sure you get the reading done. It's just a few pages. The supplemental materials are only the video clips. We no longer have a virtual lab for this lab because we had one and it was running Flash. And Flash is no longer supported and doesn't work, and we've never found another virtual lab. Explain, or the learning objectives. 1, explain how to properly collect throat and urine specimens for routine culture. 2, list common bacterial pathogens isolated from throat and urine cultures. 3, know the culture media used to cultivate these pathogens. 4, recognize which tests are used to identify throat and urine pathogens. And then 5, understand the principles and how to interpret the following tests: the catalase test, coagulase test, and the oxidase test. We will briefly talk about the XV factor testing, but I'm not going to ask you to understand it or interpret it, other than maybe, and I don't think so. There might be a question about it in the lab, but beyond that, I will not ask any quiz questions. Any question about the learning objectives? Okay. Let's move on. Let me get that coming up here. So once specimens are received by a lab, they're logged and processed in a timely fashion. Specific culture media are inoculated and incubated depending on what the clinical specimen is. We're going to talk about throat and urine specimens, and below is an overview. Don't ask me why that came up. Hopefully that'll go away. We're going to talk about the culture media used to cultivate common bacterial pathogens isolated from throat and urine cultures. So for throat, we only have 2 culture media, mainly blood agar, which is used with adults and then children and infants. And that is for testing for the presence of Streptococcus pyogenes. We also use chocolate agar primarily for testing for Haemophilus influenzae, which is seen as a pathogen in the throat of young children and infants. So not even older children have Haemophilus influenzae, or normally do not have Haemophilus influenzae, but very young children do, and then infants. And then for urine specimens, we have at least 2 media we grow the urine culture in, and that is blood agar, and then one of the 2 others. You can either grow it on MacConkey's agar or EMB agar. For blood agar, we're looking for all of these species, particularly Streptococcus, Enterococcus, and Staphylococcus, as well as the Enterobacteriaceae, such as E. coli, and the Pseudomonaceae, such as Pseudomonas aeruginosa. All of those will grow on blood agar. On MacConkey's agar and EMB, it only grows the gram-negative, such as the members of Enterobacteriaceae and the members of Pseudomonaceae. Obviously, the fact that we're growing it on MacConkey's agar and EMB agar should let you know that most of, or the majority of the urine pathogens are gram-negatives. And that's why we grow it on MacConkey's or EMB agar, is to help look at the gram-negatives. Any question about any of that? But for a urine culture, you always grow it on blood agar, and then one or the other, either MacConkey's or EMB. Throat cultures happen when somebody has a sore throat and the doctor looks in the throat and sees white patches on the inflamed tonsils or in the back of the throat. The white patches are indicative of a bacterial infection. If the throat is just red, that is indicative of a viral infection. The most common isolated bacterial pathogen in a bacterial throat infection is Streptococcus pyogenes, which has other names such as group A strep and gas. I don't use those other names. I use the the scientific name where you're actually naming the species. So strep throat is done on either a rapid testing or a throat culture. We're going to talk about the throat culture, where you swab the back of the throat or the tonsils where there's redness and or white patches. You swab the back of the throat or the tonsils. You do not want to touch the swab to the sides of the mouth, the tongue, or the roof of the mouth. Patients that test positive for Streptococcus pyogenes are placed on an antibiotic therapy. That's because patients have a hard time removing Streptococcus pyogenes. Streptococcus pyogenes has a capsule around it, and that capsule helps prevent our immune system from removing Streptococcus pyogenes. So if any of you have ever had strep throat, you know you usually have it for several weeks or a month, and it doesn't get much better. It doesn't go away. And that's because your immune system can't fight it off. And then you go in, tell the doctor, and they test you, and they come back with strep throat. And you know that's actually a serious disease. Nowadays, because of the use of antibiotics, Streptococcus pyogenes doesn't often go beyond a sore throat, but it can infect the blood and then cause very serious diseases, which are very uncommon nowadays. And I think two of them were scarlet fever and maybe red fever or something like that. I don't remember the other one, where the bacteria would go around in the body and then do serious damage, including damage to the heart, like the heart valves. Those cases are very rare now because, like I said, most children or adults who get Streptococcus pyogenes, you receive antibiotic treatment, and then it doesn't get worse than strep throat. Most of the cases that happens, not all cases. All right, Streptococcus pyogenes is fastidious, meaning it will not grow on general purpose media. The media has to be supplemented with 5% sheep's blood to facilitate its growth. And this we call an enriched media or enriched general purpose media, meaning an enriched media. A blood agar, also called BAP, is inoculated, and then the plate is examined for small, clear colonies which are usually whitish, and then they have a clear zone of hemolysis around the colony. Let me blow this up so you can see that better. It's a little slow, but my computer is probably still processing the Zoom session from the lecture. Anyways, there's the bacteria colony, and you can see it has a clear zone where there's no blood, no red of the blood around the bacteria colony. That's because this is Streptococcus pyogenes, which hemolyzes the blood completely, meaning it's beta hemolysis. Let me shrink that down now. So when we streak the throat and then grow it on blood agar, when we see beta hemolytic colonies, they could be Streptococcus pyogenes or some other bacteria that results in beta hemolysis. Only 2 biochemical tests are required to definitively prove that it is Streptococcus pyogenes. You run the Cadlase test and then the basotracin susceptibility test to prove that it's Streptococcus pyogenes. The Cadlase test just looks for the presence of the enzyme Cadlase, and if Cadlase is present, it will convert hydrogen peroxide into water and gas or oxygen. That oxygen creates bubbling, and then you look for the presence of bubbling, and then you say it's Cadlase positive. Streptococcus and Enterococcus are the species or the families of bacteria that can grow in oxygen but are Cadlase negative. Most species that can grow in oxygen are Cadlase positive. So if the bacteria is beta hemolytic and then Cadlase negative, it's likely to be Streptococcus or Enterococcus. You then run the basotracin sensitivity test, and what you do is you take the bacteria from the blood agar and streak it in this region of another blood agar plate, and then after about 5 minutes of streaking the bacteria, you add a disc containing basotracin, and then you put the plate in the incubator, incubating it upside down for about a day, no more than 2 days. And then you look to see if the bacteria is sensitive to basotracin. If the zone of inhibition is something like 14 millimeters or greater, then the bacteria is sensitive to basotracin. And that means it's, this says group A strep, I'm going to say Streptococcus pyogenes. And if the bacteria is not sensitive to basotracin, then it is not Streptococcus pyogenes. It's some other member of Streptococcus or Enterococcus because it is Cadlase negative. Any question about any of that? All right, another organism that is clinically significant when isolated from the throat, particularly a throat of an infant or a small child, is Haemophilus influenzae, a gram-negative coccobacillus. Haemophilus influenzae is the most common cause of ear, eye, and sinus infections in babies and young children. Haemophilus influenzae is fastidious, like Streptococcus pyogenes, but it will not grow on blood agar. It actually requires the presence of the V factor, NAD, and the X factor, HIMIN, which are in blood, but blood blood agar plates cannot be lysed. The blood cannot be lysed by Haemophilus influenzae. So if you were to grow it on the blood agar plate, Haemophilus influenzae will not grow. And to get it to grow, you grow it on chocolate agar, which is the same thing as blood agar. Let me see if I can shrink that and that'll disappear. Not sure why this is all messed up. There's the catalase test. Oops, what happened to my picture now? And that should be over here. I don't know what happened to this. Oops, there I lost that too. Oh, well, I'll just have to remember this. Come on over there. I don't know what happened to that. All right. Anyways, Haemophilus influenzae, if you streak it on blood agar, it will grow because chocolate agar is the same as blood agar, except the blood is added when the agar first comes out of the autoclave. And the blood of the agar is so hot that it lyses the blood, making the media not look reddish from the blood, but actually looking brown. That's why it's called chocolate agar. Let me blow that up a little bit. The only way you can get Haemophilus influenzae to grow on blood agar is if you streak Staphylococcus aureus this way, a line of it on the blood agar, and then streak Haemophilus influenzae perpendicular to it this way. Haemophilus influenzae will not grow away from the Staphylococcus aureus. Let me blow that up a little more. I'll have to make sure I don't save this when I get it. But Haemophilus influenzae will grow really close to the Staphylococcus aureus. The reason is that Staphylococcus aureus is lysing the blood in this region, and so Haemophilus influenzae can grow in this region. Any question about any of that? Normally, we don't grow Haemophilus influenzae on blood agar because we don't streak both Haemophilus influenzae and Staphylococcus aureus on the same plate. Any question about any of that? All right. All right. Urine cultures are used to diagnose urinary tract infections or UTIs, which normally are more common in females than in males, simply because the female urethra is smaller in females than in males. Bacteria can travel to the bladder through the urethra. The genital area is usually cleaned prior to collection of a urine culture, and then the patient begins voiding, and then they catch urine midstream through the urine. So not at the start of urination and not at the end, in the midstream. And this we call clean catch midstream, involving collecting the middle portion of the urine. That way there will be fewer bacteria coming from the urethra, because the urethra in both males and females is colonized by bacteria. And so there will be bacteria in a clean catch midstream. You can also collect urine by putting a catheter into the patient. The trouble is the catheter goes up the urethra, and that can put bacteria from the urethra into the bladder, and then you sample that bacteria in the catheter. But there's fewer bacteria if you use a catheter to collect the urine in the clean catch midstream. And of course, with the clean catch midstream, there's fewer contaminating bacteria than if you just voided normally. Okay. The best way to do it, which patients do not like, is a suprapubic aspiration, where you put a needle through the abdominal wall. Obviously, you degerm the area you poke, and then poke the needle into the urethra, and then sample the urine directly in the urethra. That should give no contamination of the bacterial contamination of the urine. So once the specimen is received in the lamp, 0.1 milliliters of urine is streaked on the plate. That is 1 microliter. There we see a, let me blow that up a little further. There we see, excuse me, a tool for placing 1 microliter or 0.001 milliliters of urine on the plate. And you just take that and you spread it on the plate. That's a blood agar plate. And then you streak also with a urine culture. You also streak it on either a McConkey's agar plate, a MAC plate, or an HE plate. Here they're doing a McConkey's. Interpretation for a positive urine culture is multifaceted. Some factors that need to be considered is the type of collection. Voided, even the clean catch midstream, will give more contaminating bacteria than the catheterized specimen or the suprapubic aspiration. The gender of the patient has to be considered. Males have fewer contaminating bacteria than females in the clean catch midstream. And then the number of colonies per mil. And then the presence of polymicrobial growth. If there's multiple organisms isolated, it may indicate poor collection technique. On the other hand, if there's one or a few species isolated, then that probably was good collection technique, and usually you will have several species from the clean catch midstream, but the catheterization would have one or only two. All right, some tests you run on the urine specimen that comes up. Obviously, if it grows on McConkey's, it's gram negative, and everything should grow on the BAP, blood agar plate, both the gram negative and the gram positive. Some tests we run are the coagulase test. The coagulase test looks for the production of the enzyme coagulase. It's used to differentiate Staphylococcus aureus from other members of Staphylococcus. The other members of Staphylococcus family are coagulase negative. Staphylococcus aureus is coagulase positive. If you have coagulase enzyme, what happens is the bacteria can convert fibrinogen into a fibrin clot, and then the plasma will begin to clot, and that can help protect Staphylococcus aureus from the immune system, especially when the Staphylococcus aureus has first invaded the host and is at low numbers. There are two tests, the rapid slide test, where you simply look for clumping of the plasma in the slide because the coagulase creates the clumping, or you can do the tube test where the coagulase causes the plasma to clot, clump, and then if it's a negative coagulase, the plasma will stay liquid. most common bacteria isolated from a urine culture is E. coli. And so we have to have some tests for looking at the family Enterobacteriaceae. Staphylococcus aureus is a concern in a bladder infection, and that's why we might run the coagulase test if it grows on blood agar, but not on MacConkey's. It might be Staphylococcus aureus. If it does grow on MacConkey's, it could be E. coli. And so what we want to do is separate the members of Enterobacteriaceae, which is the family E. coli belongs to, from the family Pseudomonaceae. And an easy test to run that is the oxidase test. The Pseudomonaceae family are oxidase positive. The family Enterobacteriaceae is oxidase negative. Some other possible tests to separate the two families, Pseudomonaceae from Enterobacteriaceae, is the Enterobacteriaceae is gram-negative bacteria. All the members ferment glucose, and they're all facultative anaerobes. The Pseudomonaceae are all oxidase positive. None of them ferment the sugar glucose, and they're all aerobic. So they're aerobes. They're not facultative anaerobes. The oxidase test is actually looking for cytochrome C, which is an enzyme in the electron transport chain, and we just talked about that today in the lecture. So the oxidase test can differentiate between the Pseudomonas, such as Pseudomonas aeruginosa, from the members of the family Enterobacteriaceae. If the oxidase test is positive, it'll show blue like that or violet, and that would be oxidase positive. The oxidase negative would be where the area does not turn blue or purple. All right, any question about any of these tests? So in summary, we have two cultures, the urine and the throat cultures, and microbiologists are trained to isolate and identify pathogens present in both. So in laboratory 7, you will not be physically performing any of these activities. However, you will be responsible for knowing how to perform them. We have some videos for you to watch, one on the catalase test, one on the coagulase test, and one on the oxidase test. And then we have some questions for you to answer. Let me go to the worksheet. Identify the principles used to detect the presence of catalase, coagulase, and oxidase. And one of these three enzymes will be the answers for the first 3 questions and really for the first 4 questions. So what enzyme converts fibrinogen into fibrin? And then 1B, name an organism that tests positive for this enzyme. And when I say organism, I mean micro. This is microbiology, so you cannot say humans have the enzyme. You must give me a microorganism. 2, what enzyme detects the presence of cytochrome C proteins in the electron transport system? Name 2 organisms that test positive for this enzyme. 3, what enzyme hydrolyzes hydrogen peroxide into non-toxic products of water and oxygen? And then name 2 organisms that test negative for this enzyme. 4, which rapid test is used to differentiate between A, Staphylococcus and Streptococcus? Give the answer here. And then B, Staphylococcus aureus from other Staphylococcus species. Give the answer there. C, Enterobacteriaceae. from the Pseudomonaceae. You give the answer there. And then 5, concerning specimen collection for routine throat culture, what areas of the throat should be swabbed and what areas of the mouth should be avoided. 6, what is the most common cause of bacterial pharyngitis? That is a bacterial sore throat, and you must give a bacterial answer. 7, name a species. 7, what test is used by microbiologists to differentiate between beta hemolytic streptococcus from other beta hemolytic streptococcus species isolated from the throat cultures? In the lab, we mentioned catalase. However, we're saying it's streptococcus from other streptococcus. So this already we know is catalase negative. So catalase is not the correct answer. A, answer the following concerning a child's throat culture. A, state 2 non-selective agar plates that are inoculated. And B, what organism can be isolated on CAP but not on BAP? 8C, you do not need to answer. That's why it's crossed out. 9, list 3 methods that are used to collect urine for routine culture. And it should be one of the methods listed in the lab. 10, list 3 reasons why urine specimens should be rejected for culture. That's discussed in the lab. 11, how much urine is placed on the surface of the blood agar plate on a urine specimen? And 12, what is the most common bacteria isolated from patients with cystitis? That is a bladder infection. So list a species there. 13, identify the Gram reaction, that's the Gram stain, and the cellular morphology, the shape of the cells. You should be able to find the answer in this module or in the appendix of this module. And I'll show you the appendix in just a minute. But give me the Gram reaction and the shape of Staphylococcus saprophyticus, Escherichia coli, Pseudomonas aeruginosa, Streptococcus pyogenes, and Haemophilus influenzae. Let me just briefly show you the appendix. At the end of the lab module, we have an appendix talking about some gram-positive bacteria, specifically Streptococcus, the beta hemolytic streptococci, and then Haemophilus, like Haemophilus influenzae. And then I've added the coagulase test. Any question about any of this lab? All right, if not, that's it. I will be here until the last student logs out or until 8 o'clock to answer any questions. Go ahead and get started on the lab.